Hi everyone. In this video, we will explore a new tool for single cell data annotation, that is CellLlama, and it is more of AI based tool. Okay, AI assisted, we can say single cell annotation method. So far we have seen there are, we talked about marker based annotation approach, and we explored some tools like cluster mole, SC type, easy cell type. Then we moved on to the reference based annotation like, single R, cell type is those type of thing. And the third category is very recently with the evolution of AI. So we have been seeing, different LLM models using that can we, try to annotate the single cell annotation. So that's what we will be covering. Okay, so of these, CellLlama is a very recent paper, so they are published in November 2025. And yeah, so this is a nice tool that allow us to annotate single cell data, cluster data, by giving some marker genes. That's all it will be able to do that. And the name is like cell embedding leverage language model abilities. Okay, so basically in short, right, what it does, like, it takes our cell to gene expression. Okay, so this is the usual matrix, right, where each row is equal to one cell, and let's say these are the genes. So what it does, like, it tries to find out the way, for example, we do the differential gene expression cluster 1, right? So you will get some top marker genes, right? So you can create a sentence like, top genes are, let's say these are the top marker genes. So you are creating a sentence. Okay, so top genes are, let's say top 20 genes you write, followed by, you can say, let's say the cell expression, for example, the expression of this cell is, for example, sorry, my bad, FTH expression in this cell 1 is, what is the expression value, then what is the that genes expression in that cell number 2. So basically what you are doing, you are simply creating a text format. So that means, let's say if you're considering 20 genes, so first you say which are the top genes in the descending order, and then you also pass the expression values, as well as what you also can pass the conditions, like, what cell, what tissue it is, those metadata you pass. For example, you can say here additional after these genes, right, you can pass like condition of this cell is severe COVID-19, or condition of this cell is control treatment, like that. So that means pass, convert all the information that you have in the form of a very simple text, because this large language model, they understand text, right? They have been trained against a large amount of human corpus, human data corpus, right? So that means they understand they have already read so many biological paper, like, all the articles they have already trained with that. So they understand that, they have that knowledge. So basically we are feeding this text, and this text will be actually indirectly, you can think how LLM works, right? It in that large language model space, it tries to query, so it will always predict the next token. So based on the similarity, it will try to predict. So what we are asking here is like, give me the best cell type. So based on that, LLM will predict. So very simple concept. You take it, convert it into a text, and then give it to model. The same thing what we can do, like, you can, no need to use this tool, you can directly generate this text manually, and you just upload to ChatGPT, or let's say Cloud, or let's say Gemini, and tell that, can you suggest the cell type with the reasoning. So now this large language model, although they are very general, but at least they will give you some information to start with, to begin with. So that's what the, this is very simple way of AI based annotation. Also, I have created a separate series also on AI based, how you can do the single cell analysis, but that was more towards using Claude to do the single cell analysis. But the same can be, you can use the same concept to do the annotation as well. But in short, you take the genes, you just submit to any LLM model, and then you will get the annotation. But the same thing is done behind the scene by this CellLlama tool, but it does something else. Okay, so that I will just quickly explain how it does by going through paper. So basically what it does, like, it's a framework that leverages the language model, it's GPT, those kind of language model, but to transform the cell data. What is the data? Cell by gene, sorry, gene by cells matrix into sentences. That encapsulates not only the gene expression value, the matrix, but also encapsulate the metadata, the cell's metadata, right? What the cell is, everything. So that means enabling the universal cell embedding. Okay, so that way you create this kind of sentence, and it will pass to the transformer model, and you ask the question, and it will give you the response. Okay, And the good thing about this tool is like it works with local Ollama model as well, so that you don't have to pay for running it. Okay, alternately, if you want, because this Ollama model will run in on your local system, which is very slow. Alternately, if you have some money, you can go for a paid account, OpenAI, like that you can, and then you can submit the model, that GPT model, the query, you will get the answer. Okay, so that's what we will be looking and doing some practical how this tool works. Yeah, but in short, what it does, like, just quickly overview, like what it does, like it will convert cell to sentence transformation. That means it converts the single cell data into natural language sentences. So basically what it does, sentences are built by listing the top k expressed genes in the descending order, top genes. And also you can attach the metadata, like, for example, this tissue of this cell is lungs, like this. So finally you get a entire sentence or entire query, we get it. So now then what we do, we just against it ask the questions to the LLM. So behind the scene there is embedding is there, and the same thing will work like the tokenization embedding, how the LLM works, right? The same thing will happen, and based on that it works. And this one is, it already leverages the pre-trained sentence transformer like all mini LM to map the cells into this high dimensional space without need to train a model from the scratch. Basically what it is saying in the sentence, like it uses the existing model, this embedding models it uses to do that. Okay, so don't worry about the much behind the scene what is happening, but in short, take your genes, convert it to a text and give it to LLM, it will give you the answer. Okay, and this tool has already done the benchmarking against very popular GPT tools like a single cell GPT and gene former. These are the alternate LLM based tools in terms of accuracy, precision, recall, and it was very good actually. Okay, so yeah, and the main thing what I liked about this tool is this thing, it works with the local Ollama model. So let us free, I will show you both. This one for OpenAI I cannot show because for that I might need some subscription. My subscription is expired, but I will just show you the command that you can try it out. Okay, so before that let me show you the paper, original paper. So okay, so you can go over here, directly type CellLlama in GitHub. So you will end up to this repository, and yeah, so it supports both R and Python based. So I will show you the R based, and then we will move to the Python based quickly over you. And you can install this package by install GitHub. This from, you can install it, and then I will show you the local Ollama. But let's just quickly check the paper. Okay. So this is the paper, just to quickly over highlighting you. Yeah, what they did, like the same thing, they converted into cells, transformer, and then they embed that, and then you are querying against this is the query thing happens here, and you get the output, and then based on that you can do the UMAP projection. It's very, I have not read thoroughly, but that's what I get the information by superficially skimming through it. And they have, as I said, they compared other tools as well. Where is that? Yeah, somewhere they have done the tuning, fine tuning, etc. They tried. Yeah, this is what I was saying. I took the screenshot from here, like how you can embed the metadata as well. Okay. Yeah. Yeah, whenever you have time, just go through like to understand like how it is working. But yeah, so let's go to the practical. Okay, so okay, let's see. Very simple. What we will start with, first let's run the PBMC one, which already I have done it, like, but quickly let me show you, like these are all the PBMC analysis. Okay, so up to this, we have done the PBMC, UMC, everything done. Then we are doing the find all markers, the same thing, and these are the markers. And let's say it will show like how for every cluster how many markers we have. Okay, so let it run. Just a second, it will finish. So let it run. Meanwhile, I'm telling you like what we have to do. We have to load these libraries, and the command is very simple. This much only, So this is the command. That's very simple. CellLlama, and here we pass the marker list. Okay, I think it's finished. Okay, so where were we? Okay, so we have gone the all markers, right? So these are the markers that we got, and this is the cluster wise marker. So cluster 0 we have around 207 marker, cluster 1 267 like that, and cluster 6 has 81 markers. Cluster 6 is this one. Okay, so now these are the total markers. These are not the top marker. So let's load this library CellLlama and some other libraries. This is the same plot. So now let's do the top markers, and we need to split that into cluster wise. Okay, so that's what I have done that. So if I just show you what it means, let's do the view. You will see that this is the cluster, the list actually. So cluster 0, these are the metrics. Okay, and these are the genes we have. Cluster 1 is the matrix, like that. So basically what we did, top markers the data frame that is split into different cluster wise. I have split it. Okay, because it needs a list of marker, right? So that's why we have to pass. So now here you say that this is the list, total genes 20 you take, and you give the seed is like, so that for reproduce every time you run it will work that way, and you can choose a model. Okay, now as I said, it runs with both local Ollama model and OpenAI model. Okay, when I say Ollama, right, so you can just go to ollama.com, and here this is all the free models. So you can install first the Ollama. Okay, so I think this one ollama.com, you go and you can just do like this, PowerShell, or you can download locally and it will install it. And once it is installed, right, so you can pull many models. Okay, so I'll just show you in my local system. I have Ollama already, I have installed it. So when you just type Ollama list. Okay, so it will show you all the models that you have. Okay, so like I have installed this is the model you should use, GPT OSS, which is the finest model from OpenAI that you can freely use locally. And this is GPT OSS, means it is reasoning capability, because it can think, and based on that it can plan, and it can execute. So this is more powerful model, but the problem is like its size is on 13 GB. So depending on your laptop size, RAM and this thing, it will be work. Otherwise, now let's do, for initially you can go with this local 3.2 model, which is a 2 GB, which will run fast. Okay, so just you have to remember this name, which you will need to pass over here. Okay, so here I can say that this is my local Ollama model, and my Ollama is running on this port. Okay, so then when you install it, by default it will run in this 11434 port, and so this is the Ollama URL, you can say. That means this is the local host, it's running on your local system, right? That path you have to give, and you can say this is the model. Here you can pass, as I said, GPT OSS, but it will take some time to run it. And yeah, and this is the main thing. Okay, the base prompt. So that's where you are telling this kind of system prompts to an LLM, and here you're saying that, hey, act like an expert immunologist and give me the cell type annotation for this cluster. Please reply the final cell type association and nothing else. If you're not sure, just level unsure. So this is what I'm instructing the large language model to give me the response. Okay, and temperature you can change. Low temperature means like very perfect answer, and if you want a more diversification, more creative answer, then it will more diverse, I would say, not creative the right word, but yeah, if you want a more diverse answer, then you can go with increasing the temperature to see. Okay, alternatively, if you have the OpenAI model, right, so yeah, so in this case, I think this will not work. You have to give this OpenAI key. Let's say you can define your OpenAI key. But I'll tell you, let's go through this Ollama one. Okay, so just simply you run it. It will take some time to run. Let me pause my video meanwhile. Okay, so it's running one by one cluster. So the first cluster it ran, it says response T cell. The second one is again second cluster it is running T cell. So you just have to give, that's what list of this marker, right? Take that marker list, break down into, split into a different list. And so now the third one it gave B cell, fourth one NK cell, like that it will try to do. It is very fast because I'm using a very smaller model, but GPT OSS, if you do, it will give more this thing. Okay, so meanwhile let's explore this function. Okay, so you can go to packages, and you can go to CellLlama. So, and within that we have other function as well, I will show you, but main thing is CellLlama. So here you can give get reason equal to false or true. Okay, by default it is false, but I will show you how the reason, if you say get reason equal true, it will also give the reasoning along with it. Okay, So yeah, so let's do one thing. Okay, let's, I'm just copying this. Okay, same thing. So storing, and here I'm saying get region equal to true. Okay, so now let's see how this one will run. Okay. Okay, what else we have? Yeah, so that's the main thing. It is there. Because I think it will take some time because it's reasoning, right? It has to fetch back the reason. So let's see. So it has given the reason, based on the provided description, annotation T cell is likely due to the presence of several genes that are characteristics or associated with T cell biology. Here are some possible reasons for the annotation, like cytokine receptor, transcription receptor. So like that, it has given the first response, okay, with the reason. So that's what, it will take some time because for every cluster, it will now give the region, okay. So I stopped it just to, I wanted to show you, you can try with the region equal to true here, okay. But now let's say we have this resolution res vector that stores this one, okay. So now if I just see the result structure, okay, so it's a huge data set. So let's do one thing, okay. I'll show it here. So how many clusters were? 11 clusters. So if you expand the first cluster, it says what is the final annotation and what is the description and the region, okay. So just let's do one thing, okay. This is the first one, and you see the final annotation is T cell, and this is the description what we got. So T cell cluster has upregulated genes, this one, okay, and the reason is null because I have reason equal to false, but when you say true, the reason that value will be stored here. Now if you go to the second one says, again the same information it is passes at T cell, and just to show you the third one, that will also get the B cell, okay. So nothing much information in the description, but it just says which all the genes that got upregulated, that considering which it tried to annotate, so that's what I think it is showing, okay. So this is what we got the reason, okay. And OpenAI will talk later. So after that, what we can do, we can get the annotation, okay, using this list, right? So you can pass to fetch the first one. And so if I just show you what I got here, the final annotation. So T cell, B cell, that for this one. So for every cluster, I got total 11 clusters, right? So I just have to transfer that. So I can say that name is equal to this one. And if I just now do this one, so that means 0 is equal to T cell. Cluster number 1 is T cell, cluster number 2 is B cell, like that. And next I can use the same procedure like rename identity to rename it, and I can draw the dimension plot. So now you can see that this is the LLM based annotation what you got. It's like T cell, helper cell, it's like that we are getting it here. And it has a beautiful thing, like same information if you want to store in a markdown format, it will create an HTML report as well, okay. So already I have downloaded it, so I'll just show you how the report will look like. So this is the cluster 0, the what we have shown right in the str, so the same only it is say, but if the reason is that those value will be also populated. So at least this is an HTML report format. This looks good to you can identify which gene it is, and the final, as well as you can get the answer as well why it clustered that way, okay. So the second approach is like, as they said, it's about the OpenAI based, right? So here we need to, so you have to go to your this one, platform.openai.com. So this is where you have to sign in and create an API. For example, you can go to API keys and you can create a new secret key. And but yeah, you can go to usage, right? Not usage. So this will, if you have to credit by credit, so for example, I think minimum $5 if you credit, so that will be, I think, for 1 million token or something, that will be sufficient. So okay, I finished that, so I don't have right now. So but yeah, so as I said, you get the API key. You can just go to here, give some name, dummy name, and you can say create a secret key. So it will create this key. You just copy it and come back here and just say that you know let's say OpenAI key is this one, okay. So like that you can create, okay. I'm just again So now this one you have to pass use OpenAI equal to true, and you have to just pass the key. Okay, when it does, what it will happen, this information now it will submit to OpenAI platform, and this LLM model, the chat GPT, or that GPT-5 model, you can also choose which model. And then I think here we have to specify what model, GPT 4.0 mini or 5 you want. You can use that model, and you will get a better response, because these are the older model, right? So you will get more easy response as well. Okay, so in short, right, yeah, that's all. Is a very interesting tool and very simple, I would say, compared to other tools. Like I have already, I will try to explain that in another video, like Cassia and all those are little complex tools, but this is a very very simple tool. Just one line of code, you just pass this thing, gene, and it will be give you high information. This is very similar, as I said. You just take the genes and give it to ChatGPT latest, or Claude, or any AI assistant to help you annotate as well. Okay, so with that, yeah, I think uh, we are complete with this one. Maybe we'll move to the next tool. I will have probably will go with this LLM cell type. So that is also another LLM based tool that we will go. Okay, so I hope this gave you this gives some idea about how easy it is to use AI based tools, but you have to use a combination of two. Sometimes the old tools works better, because that's where the statistical text confidence is there, but these are all about text based, right? So that's why you have to be very very careful using AI based tool. So alternately, as I said, I have created a video, right? So that's where I tell like I have shown like how you can use Claude to update the skills and whatever the tools we use, right? Like cluster mole or single R, everything. So you can create a skill file and tell the AI to do the annotation for you. So now the AI can use that knowledge plus the domain knowledge it has, and together it can combine to give a powerful output. So I would say always use a hybrid approach, and so to get a more powerful uh, no, answer, I would say. Okay, I hope this was useful. So do please write a feedback, like how did you like this, and yeah, I will see you in the next video. Thanks for watching. Bye.